Right, good morning, Year 9. Now, you might notice that it's a slightly different layout on these PowerPoints than previous ones. We have finished the Biology Paper 1 content, which is great to have been able to get that done in time. What we're going to be moving on to now, which will help you in all of your subjects, is I'm going to show you a really useful method to revise, and then we're going to revise the different parts that we have covered during lockdown. And then there's going to be a paper that I'm going to be sending out to you guys for you to have a go at filling in and answering as many questions as possible. And then another really good, useful revision technique, which you'll have a lot of in Year 11, is something called walking, talking mocks, where we go through a paper. So after you've had a lesson to complete your paper that I will be sending out in a few lessons' time, we'll then go through the paper and I will talk you through it, picking out the keywords and things like that. So today we're going to be revising cell structure, and then we're going to do cell division and transport in cells, and then we're going to have a little past paper questions exam on cells that will encompass those three things, and then we'll do a walking talking mock on that. Okay, so I'm going to include this link in the show my homework, which will take you straight there, but I've spoken in this slide how you can actually access this. So the revision technique that I'm going to go through is using the exam board specifications, and the specification is what the examiner can test you on. So during lessons, obviously you do a lot around the subject to help your understanding of it, but in the specification it's kind of the bare minimum that you need to know ready for your exams. Now, this revision technique can be done in all of your subjects and is a really good way. This is what I used to use when I was at school. It's a really good way of revising. Okay, so I'd like everyone to try this method out, see if it works for you. Obviously, it's not going to suit everyone. If you've tried it and it doesn't work for you and you have a different method that works for you, then obviously that's fine as well. So in order to find the specification for any of your subjects, you need to do a Google search. Okay, so you type in your exam board for that subject. So for instance, I think, don't quote me on this, the English one is the WJC. Other exam boards are AQA, which we use in science. There's EdXL, and there's OCR. So you type in whatever the exam board is, and then you put in your qualification. So for English, it might be English literature or English language. For science, it's combined science trilogy, because there's actually two ways to do the combined science. You can either do trilogy or synergy, and we use the trilogy. So I've put in the next one what you would type in to science. So you can actually try this out yourself for science. So I've got the exam board, AQA, the qualification, combined science trilogy, and then I've put the word specification to make sure that I get the specification, because there'll be lots of other bits of information the exam boards put out. Now, when you go onto it, you can click to download it, and feel free to save that specification. It is free out there for everyone. It's not just teachers that can access it. But it is a big document, and that can be a little bit daunting. So I'm going to show you how to actually use the specification. Okay, and that's what we're going to be doing in this lesson. So if you go on to the science specification, so if you either follow the link or show my homework, or go through the Google search method, either way, you should bring up the specification, and on the second page or the third page you'll have a contents. It will look like this. So I've put an arrow showing which bit of the specification we're looking at in this lesson. Now you should be able to click on that and it will take you straight to the cell biology section, okay, because that's what we're looking at. Now, if you go scroll down a little bit, it normally gives you some information at the top about the unit, what it's about. You'll see that there is a sort of an overarching section, which will be cell structure. Okay, so where the arrow is pointing, that is the cell structure. So that's kind of the chapter as you were doing classes. So what you can do, yourself a nice big piece of paper, A3 would be ideal, a couple of bits of A4 stuck together, and in the center, put that title. Okay, so we're looking at cell structure on this, so I've put cell structure in the middle. So then it breaks it down into little bite-sized chunks. Some of them are not so bite-sized, they're a little bit bigger, some of them are very, very small, but it kind of breaks it up a little bit into sections. So the first section is eukaryotes and prokaryotes. So we'll take that and I'll add it to my mind map. Okay, so I've added a little bubble saying eukaryotes and prokaryotes. Okay, so hopefully that makes sense to what I've done. So then, in that section, it tells you what you're expected to know. And all of these things we've covered in lessons during lockdown. So it's kind of putting everything you need to know in one area. And this will be really useful for when you get to year 11. So the first sentence says, plant and animal cells, eukaryotic cells, have a cell membrane, cytoplasm, and genetic material enclosed in a nucleus. Now, you don't want to just copy out the sentence because that's going to take up a lot of room. You need to take out the key information that you may not have already known or that you think is very important. So I've taken that sentence and I've added what I think are the most important bits for that sentence. So plant and animal cells are eukaryotic, so nice, concise sentence. And then from that, what does eukaryotic mean? It means they have genetic material in a nucleus. So I've taken that sentence and I've broken it down into two important bits of information. Animal and plant cells are eukaryotic, and they have genetic material in a nucleus. That's what eukaryotic means. So I've added that to my eukaryotes and prokaryotes bubble. The next one says bacterial cells, prokaryotic cells, are much smaller in comparison. They have cytoplasm and a cell membrane surrounded by a cell wall. The genetic material is not enclosed in a nucleus. It is a single DNA loop, and there may be one or more small rings of DNA called plasmids. So that's quite a lot of information. Now, you might think that having a cell wall going around it is important, but I haven't included that on my next slide, but you might want to add that. It's all kind of down to you. So what I've taken from that is the most important, is that bacterial cells are prokaryotic. Their genetic information is not in a nucleus, and they have these rings of DNA called plasmids, because plasmids will definitely be a new word that you will have come across in that chapter. Okay, so I've kind of taken out the key things that are going to be new to you. The third bit is a little bit trickier because it's kind of a general science skills. So it says students should be able to demonstrate an understanding of scale and size of cells, and be able to make order of magnitude calculations, including the use of standard form. And you can see on the right-hand side that it expects you to know centi, milli, micro, and nano. Now, it'd be really good if you don't know what centi, milli, micro, and nano. They're called prefixes. They go before a word, so centimeter, millimeter, micrometer, nanometer. If you don't know what magnitude they are, what size they are, then I would definitely suggest adding that to your revision map. I've taken something that a lot of students struggle with, even though you do it in maths, is the standard form. So I've put on here, standard form and they've given two examples of standard form. So 0.0003 is 3 times 10 to the minus 4 in standard form. I just made up that number. 3,400 would be 3.4 times 10 to the 3. Again, just made that number. It's just an example so that you know what it's talking about. So that is how you can use the specification to get all the information you need on one sheet. But we've only done one bubble. So it's now down to you to go away and complete your mind map. So I've helped you out a little bit on this one, and I've actually given you the little sections that are in the specification. So we've done eukaryotes and prokaryotes. I've had to move a bit of the standard form stuff just so I can fit in my bubbles. The next one is animal and plant cells. Then there's a bit about the required practical, which is using a light microscope. Cell specialization, cell differentiation, and microscopy. So using the equation to work out magnification. Now you can see that there's a lot of bubbles on here, and if I'd carried on the way that I had been doing it, it wouldn't all fit. But obviously I've had to do it so that you can read it, and it's on a lot smaller screen. This is why I said that A3 or a couple of bits of A4 stuck together will be really, really useful. So you're going to go away now, and you are going to complete your mind map for cell structure, trying to use the specification. And as I said at the beginning, some of it looks quite tricky. It can be a little bit daunting. Just give it a go, because I promise you, this is a really good revision technique that will help you in all of your subjects, not just science. Now, to make it a little bit competitive, what I'm going to ask you to do is, if you're really proud of your revision page, I want you to take a picture of your revision page and email it to me. So my email address is on the show my homework, always at the bottom. So email it to me, and we're going to post some prizes for the best ones. Okay, not just how they look, although that is useful. Using specific colours in certain places is very, very useful. Adding pictures, little sketches or doodles or drawings is really, really good. But also obviously how you've used the specification for it and trying to get it as concise as possible. So take a picture when you're done, if you're really proud of it. Send it to me, and we will be posting some prizes out to those that have done the best job. Okay, so that's it for me. The rest of this time is obviously you completing your revision map. I'm hoping that it's not going to take over your hour, your amount of time. But if it is taking too long, then we will give some more time in the future. So when you email your pictures in, can you also say how long it took you? Because if it's taking a long, long time, then obviously I don't want to do that each time. Okay? So good luck with that, guys. Enjoy it. Don't think of it as a chore. As I say, it's going to be so useful for year 11. Keep these posters, because they will help you a lot when you revise before your exams. Have a lovely day. Thank you very much for taking part. Take care of yourselves, and you'll hear from me tomorrow. Goodbye.